Welcome to DIME's Virtual Journal Club for August of 2023. My name is Jessie Backer. I am VP of Digital Measures and Diagnostics here at DIME, and I am delighted to have two incredible speakers here today, both of whom have been long-time supporters and champions of DIME's work. Elena Ismailova is the Chief Scientific Officer at Conexa Health, and Charmaine D'Emmanuel is Executive Director of Artificial Intelligence, Machine Learning, Digital and Quantitative Sciences at Pfizer. Elena and Charmaine will be presenting their recently published review, Digital Health Technology-Derived Measures, Biomarkers or Clinical Outcome Assessments, recently published in Clinical and Translational Sciences, along with a third author, Marie McCarthy from Novartis, who was not able to join today. So in just a moment, I will hand over the mic to Elena and Charmaine, but very briefly, some housekeeping first of all. Today's session, as I'm sure you all just saw, is being recorded. So the slides and the video, as always, will be available on the DIME website after the session. So keep an eye out for that. We always encourage Journal Club to be interactive. So what we'll do is begin with Elena and Charmaine describing the content of their paper and what led them to publish it to begin with. But after that, we'll move into a Q&A session. So at any time throughout the presentation today, please feel free to put your questions into the chat box. And when we reach the Q&A portion, I'll go through those and read them out. Alternatively, you can also raise your little emoji hand during the Q&A session, and I'll ask you to come off mute so that you can ask your question. But please do hold any of those verbal questions until we reach the Q&A. Okay, with that, I will hand over to today's speakers. Elena and Charmaine, take it away.
Good morning, good afternoon, everyone. Thank you, Jessie, for the introduction, and thank you for inviting us to speak here. My name is Elena Ismailova. I'm Chief Scientific Officer at Conexa Health, the company that provides scientific support for design of digital endpoints and also provides the technology platform to collect the data and data analysis. I'm giving you all this information so you can put the presentation in the context of my background. I'm a bench scientist by background. I started in the lab, and I dedicated a large chunk of my career to developing and validating biomarkers, first in the lab biomarker assays, and then I went full-time digital. Charmaine.
Hi, everyone. Well, thank you so much. It's such a pleasure to be here. And as Jessie said, I'm a big fan of DIME, and I've been working with DIME and Elena and Marie for a while now. It's really this is a topic that's close to my heart. So I'm a neuroscientist by training, and I've been at Pfizer for the past 6 years working with digital sciences and translational imaging to develop novel digital endpoints for use in clinical trials. So basically, I worked as a statistician together with data scientists to basically derive novel digital endpoints for sensor data and then deploy it across the portfolio. So we work with scratch and sleep, gait, physical activity. And I'm very well aware of the challenges that comes about with development of new endpoints and their deployment in trials. And that's why we're here today to have the discussion with all of you. So I'm very much looking forward to this.
So if we can go to the previous slide, before we jump into the paper itself, we would like to share with you briefly how this paper came together, because it was a very collaborative effort, and like many things in the field of digital medicine, it started with DIME. Charmaine, Marie, and I were presenting our previous DIME paper that was dedicated to BYOD option when it applies to wearables at the annual DIA meeting in 2022. We had a wonderful presentation. There were many discussions, and it was a very lively session. And one of the topics came, if these digital measures by BYOD or not, are they biomarkers or clinical outcome assessments? As we continued going through conference sessions, this topic reverberated, But it reverberated only in Q&A and hallway conversations, as you know, these are important, but it was never framed formally in the form of presentation, but people kept talking about it. And we decided, maybe it's worth putting something out there, at least our current thinking, to make people reflect on it and gather the momentum to move this topic forward because it's on everyone else's mind. Charmaine, do you have anything to add here?
Yeah. And I remember being at the actually DIME reception, and basically Marie and I and Elena were like talking, standing and saying, really, there is a lot of interest and a lot of activity in the field. Large amounts of endpoints are being deployed in clinical development life cycle from phase 1 to phase 2, 3, 4. Post-approval studies. If you look at the DIME's crowdsourced library of digital endpoints, for example, you see a lot of activity there, right? Across a lot of indications, but there's a gap there, right? The number of drugs that's really been approved with DHT data that have been used to support label claims, or the drug candidates which have been go-no-go decisions on these drug candidates that happened with DHT data is pretty small. And we know that one of the reasons is there's a lot of confusion in the field about how to classify biomarkers, how to classify these digital health technology measures as biomarkers or COAs, right? And I think that's what kind of drove the conversation to say, there's a gap, let's at least discuss it. We don't have all the answers for sure, but we should put it out there so that we can help drive this conversation.
If you are working in the field of drug development, the definitions of biomarkers or clinical outcomes assessments are very familiar to you, and this is bread and butter. But we decided to have a slide on definitions for those of you who may have heard of these terms, but may not be exactly familiar and may not remember them off top of your head. So biomarkers are indicators of a process. It can be normal, it can be disease pathology, or it can be a result of exposure or intervention. It may include molecular, histologic, radiographic characteristics. Sometimes you hear that the term digital biomarkers. The FDA is very clear in this regard, and they refer to digital biomarkers as a measurement method. It is about measuring something that is a biomarker with a digital health technology. Very importantly, biomarkers is not a measure of how an individual feels, functions, or survives, because it's a function of clinical outcome assessment. And it can be done via a report done by a clinician, by a patient, caregiver, or an observer, or it can be done based on performing a certain task. In the middle of all of these are definition of digital health technologies. The FDA definition that we provide here is very broad, but I would like to emphasize that the topic of the discussion here is a variety of digital health technologies that deals with sensor-based assessment. It is data generated by mobile sensors that are connected, so the data can be transmitted in real or near real time, and data is being processed by algorithms that converts the sensor output into data that can be used and interpreted. If you look at these definitions, you will say, well, the separation between the three categories is very clear. Where the confusion is. When we look at the next slide, it will be clear where it is. In order to show the overlap, and we as we were writing the paper, we thought, explaining this topic will be a snap of the finger. Actually, it is not. We had to spend quite a bit of time researching the topic and figuring out how to organize the information. In order to show the overlap between biomarker and COA categories when it comes to DHT-derived measures, we had to categorize of what is being measured, whether it's physiology or response to intervention in the case of biomarker, or it is how patient feels, functions, or survives in the case of COA. The important distinction of COA-based measures that it has to capture an aspect of health meaningful to patients. The measurements methods, how it's being measured, are important as well, because in the case of biomarkers, these are usually objective measures because it's done either laboratory equipment or electrophysiology methods or imaging if we look for conventional biomarkers, though it may include some degree of subjectivity because, let's say, histopathology image may be interpreted by a board-certified pathologist, so there is a human opinion here. But in the case of COA, it's usually a report because it's patient report how they feel. Clinicians report what they observe examining a patient, or it's an observer rating saying, this is how I see these patients getting functioning or able to perform activities of daily living. And very important to our conversation, how it's validated. How can we prove that this measure is fit for purpose for what we are trying to use it in clinical trials? And in the case of biomarker, it includes establishing method performance characteristic and establishing relationship with an outcome of interest. COA has similar ideas, but it's not exactly the same studies that require to validate COA because you have to establish the content validity, reliability, sensitivity to change, and usability. When we look for DHT measures, if we look for the totality and examples out there, how these measures are used, they're actually the mix of the two. It can measure physiology, function, physical activity, or response to intervention with the same method. It can be done and it's done objectively by means of sensors that makes them closer to biomarkers. But when we look at the validation, it has a lot of characteristics like test-retest reliability, usability, and sensitivity to change that make them alike COA. But at the same time, the relationship with the outcome of interest needs to be established, which is kind of similar to biomarkers. Okay, the DHT-derived measures, it's kind of the mixed bag of two. But the next logical question, why this is confusing, and Charmaine will tell us in a minute.
Thank you, Elena, for that great definitions. So as Elena was saying, right, DHT measures can be classified as either biomarkers or COA. So if we look at blood oxygen saturation, for example, by pulse oximetry, that's a traditional biomarker, right? If we were to use sensors and apply them on patients and have them do an instrumented performance outcome assessment, so for example, a 6-minute walk test, that is a COA because it's telling us about how the patient is functioning, right? Another example would be reduction in mobility. So if we look at gait or physical activity, right, the reduction in mobility in heart failure is predictive of mortality. So actually, mobility can be seen as a biomarker supported the surrogate endpoint. But Mobility is also important to patients. It's a meaningful aspect of health. We can look at how changes are meaningful. What is the meaningful change in mobility? And as a result, it can be seen as a COA assessing the patient capacity. So we can have multiple examples where the same measures can actually be interpreted as a biomarker or as a COA. But there's also a risk, right? Because if the digital measures are categorized as COAs, digital measures measure a vast range of concepts of interest, right? They can measure things which are active versus passive monitoring. And as a result, it might not be feasible. In some cases, it might not be feasible to establish the meaningfulness of these measures. So for example, think about toe of angle, right? Which is one of the measures that you can extract as patients with ataxia are doing a walk task, right? The toe of angle per se might be sensitive to change. It might be seen as it might track treatment effect, but it might carry very little meaning to the patients. And as a result, it might not be easy to, for example, anchor it to a PRO or anchor it to a patient impression of change, etc. And there is a danger there because we can end up abandoning these useful digital measures because we can't establish their meaningfulness to patients. And we have seen other imaging and blood-based biomarkers that are used to support surrogate endpoints in registrational trials, and yet they carry very little meaning to patients. For example, if you look at blood-based biomarkers for diabetes, if you look at tumor imaging for oncology, they themselves might carry very little meaning, but they're still very useful biomarkers. So the other problem and the other thing that's confusing is that because of this distinction between biomarkers and COAs, it becomes uncertain for drug developers on what is the best regulatory strategy to go for with a novel digital measure. And because of and why it's complicated, because if you're trying to validate a biomarker to prove surrogacy, the studies you might need are very different than if you want to do concept elicitation, meaningfulness to change, establishing thresholds for a COA, right? And that's why when the study teams are deciding on what studies to do, what data to collect, what needs to be measured, how to establish error in measurements, change in a certain measure, if you don't have a clear guidance of what to follow, it becomes very difficult for the teams to invest in this development. The other issue that we've seen, and we outlined this in a table in table 2 of our papers, is that the terminology that's used with these digital measures can vary across geography. So if you think about digital mobility, for example, and measures of physical activity and gait, when there was an EMA qualification advice for Mobilize-D looking at digital mobility outcomes as monitoring biomarkers, so the term that was used for mobility outcome is biomarkers with EMA, whereas the same measures have been submitted in documents to the FDA as passive monitoring COAs. And that becomes very complicated if you're running a global trial, right? And you have these measures that are seen as biomarkers in one area and as a COA in others. It becomes very hard for study teams to basically make decisions on how to move them forward. So now we can go with a couple of examples, of concrete examples of how this can be manifested. Elena, do you want to start with the first one?
Yes. For instance, as you know, bradykinesia is one of the features of Parkinson's disease. And let's say we want to measure bradykinesia with a DHT. I'm not getting here into a form factor or specific measures, but it can be seen in 2 ways. If we are measuring variability of finger tapping by means of a sensor in Parkinson's disease, and we are looking here for disease physiology or seeing how it changes in the context of therapeutic intervention, it is a biomarker. On the other hand, if we are taking exactly the same measure and we're asking study participant to complete a structured finger tapping task on the smartphone to assess hand function, it becomes COA. But in the essence, it's the same test with long-term implications on what studies need to be conducted and how it needs to be validated, going back to the example where we showed in the Venn diagram, in order this to be used in support of drug development. And now Charmaine is going to give us another example that is not in our paper, but was discussed to a great extent in one of the DIMES projects.
Yeah, exactly. So basically in July of last year, DIME and the Nocturnal Scratch Project Consortium attended a CPIM meeting with the FDA about advancing nocturnal scratch as a digital endpoint for atopic dermatitis. And it was interesting because one of the topics of discussion was, can digital measures of scratch be seen as a biomarker and a COA? And I think that's very valid, right, in this case, because think about scratching, like scratch endpoints, like scratch duration, number of scratches, intensity of the scratch. That can really be used as a digital biomarker because it can help you look at response to therapeutic intervention. And you're looking at change in the select disease component, the scratching, as a response to an intervention. And you can also use it, as we traditionally use biomarkers, for patient phenotyping, for pathophysiological evaluation of the disease, to screen patients. So then it fits nicely into the traditional definition of a biomarker. At the same time, you can use it as a clinical outcome assessment because you're really capturing the scratching endpoints are really capturing how the patient behavior, right, and how they're functioning with their disease and how they're feeling about the disease, especially if you then take the COA and link it to PROs, for example. So you can use it then as a COA to support labeling claims. And it was interesting when I was reading the FDA's feedback on this, is that they actually said the tool being described by DIME does not appear to be measuring a biomarker, but the definition of biomarker or COA would depend on context of use of the measurement in a specific trial. And they also go on to say that scratch is a clinical endpoint and trying to label it strictly as a biomarker may be confusing. And for them, what's important is to demonstrate that actually scratching is telling you something about the disease, right? And that changes in the scratch, in the nocturnal scratch, are correlating with treatment effects. So you can see that this is precisely why we are here, right? The terminology and about the same measures can be different depending on context of use and why we need more alignment.
And our last slide today is call for action and what can be done in the field. Before we talk about moving forward, I would like to pause and acknowledge here tremendous work that has been done by the regulators, and specifically I'm talking about the FDA and the EMA in trying to advance regulatory guidance documents and put as much information out there as possible to clarify many questions. Especially during the time of the pandemic, if you see how much in the regulatory domain we have now compared to early 2022, it's a huge difference. And it definitely helped researchers thinking and designing new experiments as evidenced by DIME and point libraries. There is tremendous progress in the field, but there are some areas that require future clarification. Part of the problem here is that in the presence of uncertainty and some of the confusion that Charmaine shared with you, sponsors may be hesitant to put it in the clinical study protocol in the endpoints other than exploratory ones, because it requires some binding commitment. And in turn, it creates problem for the regulators, because in the absence of the data and submissions by the sponsors, they don't have the data to develop further these guidance documents and develop it further so it can enable future research. And when we want to move the field forward, we need to start with a definition. That's why we introduce these definitions in our slide deck. And the definitions need to recognize that DHTs are unique and they may be different from the old definitions that were not created with these tools in mind. These tools are relatively new, and once we update the definition, the next logical step would be to understand what is common and what is different and requires adjustments with more traditional drug development tools.
Yeah, and exactly. And then the last piece actually is, once we outline the definitions and how we're measuring and how our digital measures are differ from these traditional biomarkers and COAs, we need to really understand what is the specific requirements that are needed when we have evidentiary submissions documents, and what evidence needs to be submitted so that we can push these tools to actually make decisions in drug development. And I think, as we outline in the paper, this can be achieved by pre-competitive collaborations, right? There is really, we can't overstate enough the importance of pre-competitive dialogue and discussions with regulators, pharma companies, HCPs, patients, academic institutions, other experts who can come together and really assess how these DHT tools, what are the new requirements for these DHT tools, what can be learned from what we already know, and basically how can we pave the way to the future. So I think that's really important.
Fantastic. Wow. What an overview of a very complex topic. Thank you very much to both of you for that fantastic presentation. I'm going to stop sharing and we'll move now into the Q&A portion. I just like to stop sharing so that everyone can see each other and we can get a bit of a chat going. Please do feel free to throw your questions into the chat box if you'd prefer to do it that way. Otherwise, just raise your hand and I'll call on you to contribute. But while everyone's thinking about their questions, I will get us started. So I have a question, Charmaine and Elena, that I think seems simple, but I suspect that the answer may not be. Why is it important to make a distinction between biomarkers and COAs in the first place? So for example, why would a regulator want or need to treat these two categories differently?
I can start and see, Charmaine, if you want to add something. The distinction is important because it defines the evidentiary package that needs to be submitted to the regulators to show that a specific DHT-based measure is fit for purpose. And as we shown in the example of validation, the packages for COA and biomarkers are not identical. They have similar themes, but somewhat when you look for examples that exist for DHTs, and I think the best known examples is 95th stride velocity percentile in Duchenne muscular dystrophy. It combines the both. But is it applicable to all DHT-based measures? Do they have to follow the same path? Or some of them have to be more like biomarkers or more like COA, or the totality is required? It's not clear. And as we have shown, on example of Parkinson's disease and nocturnal scratch as a concepts of interest is very much context of use dependent. Charmaine, do you want to add anything here?
Yeah, of course. Thank you. I think that's a really, really nice outline. One thing that I would add is think about the studies, right, that clinical teams need to put together and development teams need to put together to basically validate this biomarker or this COA. And there's a lot of work that goes into understanding how are we going to develop this novel endpoint, how we're gonna validate it, and how we're gonna deploy it. And the way you think about those studies would depend on how you plan to use that endpoint, right? And that's why I think we need more clarification as where these DHT tool measures fit in the drug development process as drug development tools. And I think that would really outline, having that outline would really help us as we're basically investing into studies that we can use into evidentiary requirement submissions, documents.
Right. Okay. So the distinction and the distinguishing between those two categories is around evidentiary criteria, and of course the inherent risk involved with that, with the decision to go down one path or another. Okay.
Right. It's also about the predictability of regulatory review.
Yes. Okay. Fantastic.
Because even the FDA said many times that try to contact agency as early as possible in order to schedule some interaction with the FDA, whether it's Type C meeting, whether it's critical path innovation meeting. A package needs to be submitted to this meeting for the regulators to provide their feedback. And if the package is on the wrong path, the predictability decreases.
Got it. Yep. And all of these conversations and packages, they all obviously require resourcing. Barry, looks like you have a question.
Yeah, I just want to make a comment, partly echoing what has been talked about. But on the slide that showed how scratching can be either a biomarker or a COA, I don't see that as a problem. That's not a bug, it's a feature. And I think it actually highlights how great the digital health technologies are because you can use them in many ways simultaneously. And then I think, Charmaine, pretty much echoing what you're saying, the decision is how to validate, I don't think is all that difficult. It depends completely on the claim, just like you do in drug development. You make a claim and you develop your development program around the claim. If you're claiming that scratching is to be used to identify a change in effect of a drug, then it's a biomarker and you validate it appropriately. But if you're claiming that it is recording how a patient feels, that's a completely different question and completely different validation. But I don't see a conflict, and I also see it's relatively straightforward on how to handle those cases.
Let's dig into that. Charmaine, go ahead. Looks like
I think you're spot on, right? That like that's I think one of the powers, right? One of the opportunities actually of DHT-derived measures that it's and I think I see Jennifer here and I see Kerry, and I would love to hear also their input. I think it's a feature, not a bug. It's really great that we can use digital health tools as that we can use digital health tools to basically derive information that is going to give us so much context about like the patients and also the physiology of the disease, and you can use them in different manners. But I think keeping the context of use in mind is obviously critical. But what can be difficult is if, for example, even in the terminology that's being used, right? If in Europe you're thinking of the same measure as a biomarker, and then in another region, like in the US, you're thinking of the same measure as a COA, and there isn't alignment of how you're looking at it, right? In a biomarker, as we say, you don't necessarily need to prove meaningfulness to patients. Whereas in a COA, you absolutely need to do that with an anchoring method, with some kind of anchoring and matching to a patient reported outcome or a patient global impression of change, etc. So the requirements can be different, and I think that's where we see a little bit of confusion, like some confusion that needs a little bit more clarification, because like the qualification for both is different. And maybe it's a matter of like you qualify the same measure as COA or as a biomarker, depending on context of use, and then you submit those requirements to different paths, depending on what you plan to use it, which is what I think you're saying. Elena?
I think it's a great answer. Thank you, Charmaine.
Okay, we have a really excellent question in the chat from Victoria Hunter, but before we go there, we've also had some Carrie, Jen, anyone else want to jump in here while we're on this topic of scratch being a biomarker or a COA or a bit of both?
Definitely a bit of both. It depends, the case you want to make. And Charmaine, to your point, I think it's and Elena, you started with this in response to Jesse's question about, why does it matter? It's the evidence packet when you are using these things as medical product development tools. That's exactly the reason. I think this is also why the V3 framework is so important, because you can think about the bottom two pieces of that stack. The verification and the analytical validation for that tool will hold, regardless of whether you are making a claim that this is a biomarker or that you want to use it as a COA. Then it's that clinical validation layer that you would adjust and would be different within your evidence packet. I also think our level of comfort with the ability to embrace, Barry, back to your point, the fact that it could be either or, depending on the evidence that you're able to put together, also allows us to think about pulling these products and tools into the post-market, which I think is really important. If we are developing, measures that matter, we should think about, should we be using these as remote monitoring tools during routine patient care? If we want to start capturing real world data and evidence using these tools, we have to be thinking about the entire product life cycle. So I think it's less about a black and white decision making, but it's much more about being comfortable with a distinction and thinking about it as an opportunity to be fit for purpose in our evidence collection rather than being overwhelmed by the distinction.
Mm-hmm.
I don't really know if I have much to add. I think, Barry, way to go. I like the comment. I fully agree. I think I put that in the chat. I think a lot of the biomarkers, COAs, the new endpoints that are being developed can be used in different ways. And I just think we have to keep an open mind. I just echo everything that's said, and I further echo Jennifer. We can create all these new measures, but I think we also have to start to think about how we're going to apply these in the real world and make them tools available for healthcare providers when it's done. Because ultimately, a patient or healthcare provider really cares what it's called, as long as it helps in the diagnosis and treatment patient. So I think that's just the one thing. Even we all have to step back and go, as long as we can prove that it does what we say it does, to the level we say it doesn't really matter what we call it. So
yeah,
thanks all.
Thank you, Carrie and Jen, and of course Barry for raising this. One thing I would add, and we discussed this with Maria and Elena extensively, is kind of the risk, right? Like if we basically have a measure, and that measure, it might not be so straightforward to establish the meaningfulness to patients in that measure, but that measure is sensitive to change, is really showing you progression. It's sensitive to progression with the disease, right? The risk is you can abandon that measure. It will never be qualified if it goes down the COA path, no matter how good and sensitive it is. And that's the risk that we want to avoid, right? We don't want to end up in a situation where we're abandoning digital measures because we cannot establish direct meaningfulness to patients or like interpretability of the change in that measure to patients, right? Think about like gait speed. If you tell a patient you need a 0.1 meters per second increase in gait speed, what does that mean to the patient? But we can all agree that mobility and gait is critical to the quality of life of patients, right? So measuring gait and physical activity is important. Pushing for digital endpoints of gait and physical activity is important, but maybe they're not always going to fall very clear-cut into a COA, traditional assessment. And that is, I think, what we find confusing and what we need to de-risk.
Right. Well, whether it was intentional or not, I think that discussion has set us up really nicely for Victoria's question. Is it your opinion that measures captured by digital health technologies should be defined as a new category separate from COAs and biomarkers in terms of regulatory pathways?
I think it's an excellent question, and I will give only a vague response because the definition should be driven by data. Unfortunately, for reasons that we already discussed, we don't have very many examples of regulatory approvals out there that will kind of guide the discussion. And it's not my opinion that matters. It matters the collective opinion of the scientific community, such as DIME, what makes sense to do and what will help us to advance these tools and advance novel treatments. And this needs to be discussed collectively. We discuss in the papers some of the historical perspective and, for example, the role of NIH biomarker definitions group and how getting these definitions and defining what biomarker is and what is not, and what is a validated biomarker, really boosted the development and incorporation of biomarker tools in clinical studies. I'm old enough to remember how the field looked before all this work was published. And there were endless discussions on clinical teams. What is a biomarker? Do we need a biomarker? What constitutes a validated biomarker? Today, if you open a clinical study protocol, it will not have one biomarker. It will have multiple because the utility of these tools have been proven. And what we are trying to do with our publication is to drive the digital field to get the same level of acceptance. But before we get there, we need to do the groundwork, as Charmaine indicated, through pre-competitive work, because that's the only one possible work. It should not be Pfizer definition of a digital measure is different from Connex's definition. I'm exaggerating here, but that's what, it's an illustrative example. Charmaine, do you want to add here?
Yeah, I mean, I echo what you said, and think about why we're here and why we're at this point in time, right? Because of all the great conversations that happened over the years. If you think about like the leaps that we made from 2017 to now, it's enormous, giant leaps, right? The V3 framework, phenomenal. Like we have established, like DIME has established a playbook, which have helped so many drive the conversation and basically get us in a unified spot. I think many initiatives of DIME have focused on getting us together on definitions in very different areas, right? I think that needs to keep on happening to move the field forward and basically then discuss, okay, how are they different? How they are the same? We shouldn't be reinventing the wheel for sure, but we might need to recognize that there are some aspects which are going to be different. And I think as long as we align together on what are the aspects that are different and what's required to prove their value, then I think we'll keep on moving forward.
Fantastic. So, Barry, I'll throw to you in just a moment. To everyone else on the call, please do keep putting your hand up if you want to chime in here or add into the chat if you prefer to stay off the mic. But Barry, over to you.
Okay, well, my response to Victoria is no, I do not think it should be a separate category. In fact, I think that would muddy the waters. The fact that the DHT can be used as either provides guidance as to how it should be validated, depending upon what you want it to be. If you make DHT a separate category, we have no idea how to validate it because we haven't stated what it's going to be. So let's keep those two separate, and I think it's going to work very well.
Fantastic. Thank you so much, Barry. Elena and Charmaine, are there non-digital examples of measures? So measures that are not captured by DHTs that fall into this same kind of gray area that we're discussing between biomarkers and COAs? Or is this confusion only now arising because of the increased adoption of DHTs? Like, I guess what I'm saying is this a new problem? Or is it just putting a spotlight on a problem that may have been there for some time?
I've spent quite a bit developing and validating biomarkers. To me, it's a novel problem because we have a novel method of measuring certain concepts of interest. And because it's a novel measures, it comes with a new features that we need to understand, and we need to understand how it fits in the paradigm that was created without thinking about these measures. Barry may be well right saying that we don't need new definition, then we need to understand how the definitions that we have now work, and very important, how to apply them to real life examples that we tried to show in our paper and analyze these definitions based on examples provided in the public domain.
Harry.
I just wanted to throw out there, and I put it in the chat as well. Remember, DHTs are a tool. They're measuring something, that the measure itself. So we often combine the DHT and the measure together. The device measures this. Often the devices are just a series of sensors in development. So I think we often think we need to create this whole new category, et cetera. The measure itself, in this case, I'm going to use Scratch because it's one that everyone associates with me. Just kind of, I'm rolling into it today, leaning in. Scratching's been monitored for a long time. It's something they've done it in physicians' offices. They look at exudations, et cetera. But now we just have a new method to measure it. So now we have to go back and just say that the method that we're measuring it with is accurate. And I think we get so caught up in how we're measuring it that we forget, like Scratch has already measured. Gait's been measured, 6-minute walk test. While I'm not a fan, it's been measured. So now we're just looking at a new way to measure it. And now we have to demonstrate that that new way measures it accurately. And I think sometimes we just get caught up in that we have to link them together. And ultimately, we're measuring a concept. That concept's now being measured by a digital device. What's important is we make sure that the digital device is doing what it's supposed to be doing. The algorithm's measuring what it's supposed to be measuring. And then the third part is the measurement meaningful and or doing what it's supposed to be doing. So I just wanted to put that out there as well.
Yeah, and that's very, thank you so much. We completely agree, right? We're not talking about the devices, but we're talking about the measures, DHT-derived measures, right? And while, as you say, the concepts have been measured a long time, but the endpoints themselves, like for example, if you're looking at scratching, right? Now you can get at the scratch duration, you can get the intensity, you can get the number of scratches. You're quantifying things that you are not previously able to. Similarly, if you're looking at gait, now you can look at stride velocity, at stance time, at swing time. Those are spatial characteristics of gait that before, if you do a 6-minute walk test, you can look at the distance measured, but you're not really looking at those spatial characteristics of gait, which can inherently be a biomarker, but can also tell you information about function and might correlate with functional measures of patients. So I think that's kind of what we're talking about. But definitely, we're only focusing here on DHT-derived measures, not specific devices or technologies.
I would like to add here that the problem is not new. I completely agree with Carrie that it's a new method. And as I said, digital biomarkers does not create a new category of biomarkers. They still can be safety, pharmacodynamic, monitoring, efficacy biomarker. It's a measurement method. But it's very important to understand what measurement method can and cannot do. And this has been solved in the biomarker field, and there is another DIME paper that we did together with Jesse under the umbrella of DIME Research Committee that actually grouped different technologies by the level of quantitation that they can do. Fully quantifiable, semi-quantifiable, quasi-quantifiable, and qualitative. And having understanding what needs to be done for each category, what can be done and what cannot be done, and what are the data limitations was a major driver. And this coupled with where a certain measure supports an endpoint, whether it's primary, secondary, or exploratory, defines the fit for purpose and how many experiments to be done. And this is what can be accomplished through pre-competitive work. Once we have a clear understanding of categories and how they fit into the context use and the purpose of use, will give us a way forward. The same concept of quantitative or qualitative methods used for biomarkers don't really work for DHTs because that's not how digital technologies work, but we can use it as an analogy to stimulate future thinking.
Yep, absolutely. Completely agree. Barry.
Answer your question about, is this a new problem? No, it's not a new problem, but it's a big difference. The problem is now recognized, and I think this is a major advance. As an anecdote, back in the 70s, when we were looking at lung function tests, the validation was to determine whether or not you could determine a young smoker from a young non-smoker. And the joke was, of course, you just ask them, why do you need this test? Or you smell their breath. And that type of validation approach actually mixed up the two, the biomarker and the COA, and never any discussion about it, ever. And this really has gone on for years. So it's an old problem. It wasn't recognized. I think the digital health technologies, like all of what's happening, has brought into focus old problems. And this is why DIME exists. And it really is the right tool for the problem that has been around for a long time. Finally, we're facing it.
Bit like fashion trends. we've often seen them before. All right. In just a few minutes, I'm going to wrap up with some announcements, but I do have one more question to pose to Elena and Charmaine. The paper, which I have here, because I'm still somebody that prints things out, it ends with a few problems that are, at least in theory, solvable problems. And you've highlighted a couple of avenues under which or through which we could solve those problems. So multi-stakeholder collaborations, for example, or public-private partnerships. I'm wondering if one or both of you can delve into that a little further, into how these various stakeholders might work together. So for example, what would a pharma company look to a technology company to provide, or vice versa? Like, how do you see those interactions playing out?
Yeah, I mean, I can start. So think about like what it takes to validate a biomarker in the concept of an endpoint, in the concept of the V3 framework, right? So you need verification, analytical validation, clinical validation. Now think of a pharma company. They might rely on a device company to provide documentation on the testing and the verification of the device. For example, what bench testing has been done prior to deploying it in a research study or a clinical trial. And then from us, by deploying the device in our studies or testing the device in pilot or phase 0 studies, we're helping the companies understand the success factors and the limitations, right? So there is already a dialogue and a conversation that can happen there on what's already been done, so we don't need to repeat, and then what new learnings from the studies that we run that we can bring to the table. Same goes, right, when you look at, for example, the raw DHT data. You're looking at the raw data, you're using some algorithms to develop these endpoints. If the algorithms are open source, other members of the community can use those algorithms, and then the learnings can grow and foster. So there's a lot that can happen in a community basis on a community level to basically continue to push the field forward.
I'm going to use the analogy from my previous life when I was developing biomarker assays and say that DHTs are different in this regard because the technology, it's not an inherent part of early preclinical development and maybe even early clinical development, because it's not laboratory test or imaging technology that you develop very early in the program. and later translate into the clinical. We can use, I would say, measuring TNF alpha in a mouse model. We can also measure TNF alpha in the human. We will need different set of reagents and maybe some additional clinical validation that we don't do in the mouse model, but it's translatable. DHTs are different. And that's where the power of partnership comes from, because the technology sector brings this expertise to the table, working with a pharma sponsor. Another distinction here is a role of patients. The role of patients is very important in identifying what is important to patients if we are talking about COA in the study design, and even how the technology or drug will be used in the future. And it's outlined extremely well in the new guidance of patient-focused drug development. There's a lot of wealth of information. And the unique feature of digital health technology is that it's very multidisciplinary field. And I cannot say enough that I'm so happy that DIME exists, because it brings all these multidisciplinary fields together and helps us to have a conversation like this one, which is extremely meaningful. I learned tons from this conversation. Even I'm one of the authors on this paper, because these are excellent questions, and hearing different points of view and having a forum like this is priceless.
Well, what a note to wrap up Journal Club on. Thank you very much, Elena and Charmaine, and of course, Marie, not only for the work on the paper, but also your presentation today and the fantastic discussion from everyone who was here. I also I love the fact that this paper arose from a previous paper and discussions at conferences. I mean, that's why we're here, is to kind of tackle these thorny questions. So with a couple of minutes to go, I do want to be respectful of everyone's time. Please be aware and excited for the upcoming presentation on September 6th from our colleagues working within the Impact Initiative focused on virtual first care, and specifically, really important topic, navigating reimbursement for virtual first care. We also have, speaking of scratching, a virtual journal club coming up in September, looking at nocturnal scratch, a study bringing patient input into development of novel digital measures, September 20th. So please keep an eye out for that as well. Thank you very much for joining us, for hearing and participating in the discussion. The slides and recording will be available. Otherwise, we will see you all again in September, if not before.